There is a specific intensity of aerobic exercise that produces more mitochondrial benefit, more brain benefit, and more longevity benefit than any other intensity. And most people who exercise regularly never train at it, not because it is too hard, because it feels too easy. Zone 2 cardio is the intensity at which you can hold a conversation in full sentences without gasping, where your breathing is elevated but controlled, where you could continue for a long time without significant discomfort. It is the pace that most people abandon in favor of harder, more intense exercise because it does not feel like enough, because it feels like a comfortable walk or a slow jog rather than a real workout. The biology disagrees. Zone 2 is where the most significant adaptations for metabolic health, brain function, and biological aging occur. Adaptations that higher intensity exercise does not produce as effectively, and in some cases actively prevents. This is the story of what Zone 2 training does at the cellular level, why intensity matters more than most people realize, and what 150 minutes per week of the right kind of exercise actually does to the age of your brain. To understand why Zone 2 is specifically effective, you need to understand the energy systems that different exercise intensities recruit and the cellular consequences of recruiting them. The human body has two primary pathways for producing ATP, the energy currency of cellular metabolism. The aerobic pathway uses oxygen to metabolize fat and glucose through mitochondrial oxidative phosphorylation, a process that is highly efficient but rate-limited by oxygen delivery and mitochondrial capacity. The anaerobic pathway, glycolysis without oxygen, is faster but less efficient, producing lactate as a byproduct. At low to moderate exercise intensities, the aerobic pathway can meet the muscle's energy demands. Mitochondria are operating within their capacity. Fat is the primary fuel, with glucose contributing increasingly as intensity rises. This is the Zone 2 range. Aerobically sustainable, primarily fat-fueled, mitochondria working efficiently within their capacity. As intensity rises above Zone 2, energy demands begin to exceed what the aerobic pathway can supply at the required rate. The body increasingly recruits glycolysis, lactate accumulates, and the exercise becomes anaerobic. This is the threshold, called the lactate threshold or ventilatory threshold, above which the character of the metabolic demand fundamentally changes. The specific cellular adaptations produced by Zone 2 training target the aerobic pathway and the mitochondria that power it. Mitochondria are not static structures. They are dynamic organelles that increase in number and efficiency in response to the appropriate stimulus, a process called mitochondrial biogenesis, and decrease in number and efficiency in the absence of aerobic demand. The primary molecular signal for mitochondrial biogenesis is a protein called PGC1-alpha, peroxisome proliferator activated receptor gamma coactivator 1-alpha. PGC1-alpha is activated by the cellular conditions produced by aerobic exercise, specifically by the increase in AMP to ATP ratio that signals energy demand, and by the reactive oxygen species produced at moderate levels by mitochondria working efficiently within their capacity. Zone 2 exercise produces sustained activation of PGC1-alpha at a level and duration that maximizes mitochondrial biogenesis. The mitochondria are working, they are stressed moderately, and the cellular signals for adaptation are sustained throughout the exercise period. High-intensity exercise, interval training, heavy lifting also activates PGC1-alpha acutely, but through different mechanisms and with different downstream effects. High-intensity exercise produces much higher reactive oxygen species, activates different stress response pathways, and requires longer recovery periods during which the training stimulus is absent. The aggregate stimulus for mitochondrial biogenesis per unit of time is lower than Zone 2 training despite the higher intensity of individual sessions. The practical consequence is measurable. Studies comparing chronic Zone 2 training to high-intensity interval training in matched populations consistently find greater improvements in mitochondrial density and oxidative capacity with Zone 2 training at equivalent weekly training volumes. More mitochondria per cell means more aerobic capacity. More aerobic capacity means more efficient fat oxidation, better metabolic flexibility, better glucose regulation, and greater capacity for sustained physical activity. These are the fundamental metabolic adaptations that underlie the longevity benefits of aerobic fitness. BDNF, brain-derived neurotrophic factor, is the protein that aerobic exercise produces in the brain, and it is the molecular link between physical activity and the brain changes that most directly affect cognitive aging. BDNF is sometimes called brain fertilizer. The name captures something real about its function. It promotes the survival of existing neurons, the growth of new synaptic connections, and in the hippocampus specifically, the generation of new neurons from neural stem cells, hippocampal neurogenesis. It is the primary molecular mediator of the well-documented relationship between aerobic exercise and improved memory, learning, and cognitive function. BDNF production in the brain during exercise is driven by several mechanisms. Increased cerebral blood flow during aerobic exercise delivers more oxygen and glucose to the brain and activates shear stress responses in the vascular endothelium that stimulate BDNF production. Lactate, produced in muscles during exercise and transported to the brain, directly stimulates BDNF synthesis in neurons. And the systemic hormonal responses to aerobic exercise, including irisin, a muscle-derived hormone released during sustained aerobic activity, cross the blood-brain barrier and increase BDNF expression. Zone 2 is the intensity that optimizes this BDNF response. It produces sustained lactate at low levels, enough to provide the lactate signal for BDNF, but not so much that it interferes with sustained duration. It produces sustained elevated cerebral blood flow more than at rest, sustained over a longer period than high-intensity intervals. And it produces sustained irisin release. The aerobic duration matters as much as the intensity for irisin secretion. High-intensity intervals produce acute BDNF spikes but briefer sustained periods of the driving signals. Zone 2 produces a lower peak but a much longer duration of BDNF-stimulating conditions. And for hippocampal neurogenesis, which requires sustained trophic support rather than brief acute stimulation, duration appears to matter more than peak. The brain aging data from MRI studies provides the most direct measure of what Zone 2 training does to the biological age of the brain. Several large-scale studies have measured brain structure using MRI in adults across a range of aerobic fitness levels, quantifying hippocampal volume, cortical thickness in regions associated with executive function, and white matter integrity, all of which decline with aging. The relationship between aerobic fitness and brain structure is consistent and substantial. A landmark study from the University of Illinois enrolled previously sedentary older adults and randomized them to 1 year of moderate aerobic exercise, walking at Zone 2 intensity, approximately 40 minutes 3 times per week, or stretching as a control. The aerobic exercise group showed a 2% increase in hippocampal volume, a reversal of the 1 to 2% per year decline that is typical in this age group. The control group showed continued hippocampal decline. 2% hippocampal volume increase corresponds to approximately 1 to 2 years of reversed hippocampal aging. 1 year of Zone 2 walking 3 times per week produced this reversal in previously sedentary adults. Studies examining the relationship between VO2 max, the primary measure of aerobic fitness, and brain biological age using MRI data from large populations have found differences in brain aging of 7 to 10 years between high-fitness and low-fitness adults of the same chronological age. These are differences in the structural integrity of brain tissue, thicker cortex, larger hippocampus, more intact white matter, that directly correspond to better cognitive performance on memory, attention, and executive function tests. The 150-minute per week figure, the amount of moderate aerobic activity associated with the documented brain and longevity benefits in the epidemiological literature, corresponds almost exactly to the Zone 2 training volume that produces these structural brain changes in intervention studies. It is not a round number chosen arbitrarily. It is the dose at which the accumulated evidence shows consistent benefit across multiple health outcomes. How do you know if you are actually in Zone 2? The subjective criteria are simple but require honest self-assessment. You should be able to speak in complete sentences without pausing for breath. If you can only get out a few words before needing to breathe, you are above Zone 2. If you feel you could easily sustain a phone conversation with no awareness of your breathing, you may be below Zone 2. A more precise method uses heart rate. Zone 2 corresponds approximately to 60 to 70% of maximum heart rate for most people, and maximum heart rate is roughly estimated at 220- age, though individual variation is substantial. A 40-year-old has an estimated maximum heart rate of 180, making Zone 2 approximately 108 to 126 beats per minute. The most precise method uses a metabolic measurement of the lactate threshold, the intensity at which blood lactate begins to accumulate above resting levels. Zone 2 is the intensity just below this threshold. Without lab testing, the conversation test is the most practical guide. One of the most common errors in Zone 2 training is drifting above the zone, starting at Zone 2 and gradually increasing intensity as the session continues, ending the session above the threshold where the specific mitochondrial and BDNF adaptations are maximized. Maintaining a conversational pace throughout the full session, including the later stages when it feels easier to go faster, is the discipline Zone 2 training requires. Here is the synthesis. Zone 2, aerobic exercise, sustained activity at a conversational pace, produces mitochondrial biogenesis through sustained PGC1-alpha activation more effectively than higher intensity training at equivalent weekly volumes. More mitochondria means better aerobic capacity, better fat oxidation, better glucose regulation, and better metabolic health across every measured parameter. It produces BDNF through sustained cerebral blood flow, sustained lactate, and sustained irisin release, the combination that most effectively drives hippocampal neurogenesis and the synaptic plasticity that underlies cognitive function. MRI studies show brain structural differences of 7 to 10 years of biological aging between high-fitness and low-fitness adults. Intervention studies show reversal of hippocampal decline with 1 year of Zone 2 walking at moderate volume. The 150-minute per week dose produces consistent benefits across the epidemiological literature. Zone 2 feels too easy. That is the point. The mitochondrial adaptation that determines metabolic health, the BDNF production that determines cognitive aging, and the hippocampal structure that determines memory function are maximized at the intensity that most people are tempted to exceed. Slower is the correct prescription. Not always, but for 2.5 hours per week, slow down, hold a conversation, and let the biology do what it is designed to do at that pace. If this changed how you think about what a real workout looks like, hit the like button.